My name is David Duan, and today I'll be presenting on my research on validating and quantifying mRNA expression of various glaucoma-associated genes in human donor eyes, and my mentor was Sarthak Zavuri. Glaucoma is an ocular disease that affects around 3 million patients in the United States and serves as the second leading cause of blindness worldwide. It occurs when when the trabecular meshwork is blocked, and the trabecular meshwork is located here and is made of spongy collagen fibers and can be compared to as an adaptive filter, as the less permeable it is, the less liquid that is able to pass through. This liquid is known as aqueous humor, and reduced aqueous humor drainage increases the intraocular pressure of the eye. This pressure pushes against the back of the eye at the optic nerve, causing a loss of peripheral vision, as seen in this image right there. The current treatments include daily eye drops, and these are ineffective for a whole load of reasons. For example, they starve the eye of nutrients and cause red eye in around 10 to 50% of patients and blurred vision. There's also the issue of compliance. In around 10% of patients who are blind due to glaucoma right now, this can easily have been prevented because they did not follow the regimen even after being diagnosed with the illness. There's also the issue of pathological processes in the trabecular meshwork not properly being addressed. These medications only address the intraocular pressure of the eye and not the root cause of the genetical differences within glaucoma patients. So ideal glaucoma therapy solutions would include optimized compliance, better intraocular pressure control, and disease modification at the roots. Well, the first gene that we'll be studying is transforming growth factor beta 2. What this gene does is it reduces the TM adaptivity through stiffening of the extracellular matrix. Basically, it causes the filters to be more rigid, causing less outflow of aqueous humor. And in an enzyme-linked immunosorbent assay, this was found to be 3 times more in glaucoma patients. And next is the gene secreted protein acidic and rich in lysine. What this gene has been found in mice have been found to have a 15 to 20% lower intraocular pressure, as well as humans saw a 25 to 30% increase when inserted into donor human eye tissue through ex vivo human perfusion organ culture. So my experiment will utilize RNAscope, which binds to the specific genes and the mRNA level, basically allowing us to visualize gene expression through fluorescence, as in this image right here. So gap in literature is that previous studies have not utilized RNAscope nor duplex RNAscope to visualize the location of these genetic expression to allow us to see where these are expressed in the different structures of the eye and compare between them, such as either the retina, optic nerve, or trabecular meshwork. So, like I said before, the goal of the experiment is twofold. First is to validate the expression of glaucoma-associated genes, and then pioneer the protocol for duplex RNAscope. What duplex RNAscope allows us to do is visualize the genetic expression of 2 genes using the same tissue, allowing us to save precious and expensive human donor eye tissue, as well as compare between the two genes. Basically, we first selected 5 donor eyes, and then pre-treated them and fixed the cells and prepared tissue slides, and then we increased the permeability of the cell membrane using target retrieval. We then use amplifiers such as probes, and these target the specific RNA sequence within the cell, and the amplifiers go on top of the probes, as seen in this image here, and the fluorescence goes on top of the amplifiers, allowing us to see the expression. And here are the 3 staining that we did. Then we use image quantification. I taught myself image J, and I was able to quantify each individual dye or each individual dot of expression. I then record the data using Excel and graphed it and analyzed using graphite prism, which I also taught myself. So here are the 2 trials. First, basically in the second trial, what we did is that we used a total of 8 eyes, 4 glaucoma and 4 normal, with the experience that I got from trial 1. In trial 2, what we also did, as seen here, is we also zoomed back from the image so we can get a more representative image of the whole entire eye. And as you can see, this is ciliary body. You can see the red is the spark, the green is the TGF-beta2, and the blue is the DAPI cell expression, the cell nuclei, which stands for each individual cell. It's clear in the optic nerve. It is basically pretty highly expressed, in both groups. And here's the retina. You can see it's medium expression in both groups. And here's the trabecular meshwork. It is pretty lowly expressed. So basically what we found is that TGF-beta2 and spark are highly expressed in the ciliary body and optic nerve, as seen in these 2 graphs here. The graph on the left is trial 1, graph on the right is trial 2. As you can see, the optic nerve and ciliary body has significantly higher expression. This number is the copies of mRNA per cell. So what we did was we found the fluorescence per cell divided by the divided by the cell nuclei to find around 1.493 copies of mRNA per cell for Spark, and this is pretty significant. This is data that that is very significant and very promising. However, there was low expression in the TM and retina, and after performing a t-test, there was no difference between the two groups. And this also shows the difference between the proteins and genes. My data does not correlate with the previous studies, however, because previous studies used proteins and mine used genes. So that could be a potential issue with a different methodology and different focus of our experimentation. So applications that my experiment proved the effectiveness of the duplex RNA scope. Like I said before, we were able to save precious donor eye tissue by using two by analyzing two genes for the same tissue, as well as I was able to use data for quantification to see the specific portions with so basically instead of using instead of using a PCR and lyse the whole entire tissue, we're able to see the data for quantification within each specific structure. As a result, we can target glaucoma associated genes as an ideal glaucoma therapy potentially in the future. So possible limitations is that there's possible damaged tissue, as these tissues could be damaged through transporting or storage, as well as a small sample size of only around 8 patients or 8 samples for being glaucoma and for being normal. This meant that this small sample size could have not been representative of the whole entire sample or of the whole entire population. Further testing is needed to see more glaucoma-associated genes, such as the ROROC pathway, as well as analyze genetic silencing, and these could be possible future treatments such as siRNA or ASOs. What these treatments do is that they basically silence or they basically lower the expression of these genetic factors for potential therapeutic ideal glaucoma treatments in the future. And yeah, I would like to thank my science research teacher, Mr. Menig, my mentorship program mentor, Sardak Zaveri, and the Regenerate Mentorship Program for providing me with this amazing opportunity. And here are my citations. Thank you.